Danny Levine is hosting, and we will be talking to Chris Hart today and Rachel Groppo. Thank you.
Great. Is this working? Yeah, it's great. So first off, thanks Danny and the Global Genes team for inviting me to speak. I'm excited to share, and I think one of the things I love about this event, I've come a couple of times, is that it's sort of a catalyst, and it's just a first start in terms of making sure connections happen and seeding initial conversations. So what I hope to do today with the conversations about ASOs and siRNAs is really provide that starting point, a place to demystify what they are, how they work, and then how it connects back to what we heard in the earlier sessions associated with, how do the different disease mechanisms and different mechanisms in which disease pathology emerges, how does that interplay with how you can use ASOs or siRNAs, or as I sometimes like to think about them, oligonucleotide-based medicines generally to help develop therapies. So I'll kick things off, so first off, I think the most important thing is to understand what they are from a foundational basis, and I'll start with key takeaways, and then as we go through things, hopefully you'll remember the key takeaways. I'll come back to these at the end. But effectively, when I say oligonucleotide-based medicines, I might refer to them as OBMs during the talk, and that's just a way for us to refer to this class of medicines. And when I think about them, these are synthetically created, chemically modified DNA and RNA molecules. And this would include ASOs, which are antisense oligonucleotides, or short interfering RNAs, siRNAs, guide RNAs, which are also synthetically created, or guide DNA molecules. And at the sort of foundational level, they're just chemically modified polymers that are made out of nucleotides. And when we make them into drugs, we have to modify the sequence and the chemistry to make them have favorable pharmacology. When we think about how they act, this is where it becomes really important, and it connects back to what we were learning about in terms of disease mechanisms, because OBMs act as they bind to RNA and DNA often in the cell, so that they can actually act as drivers of functional change of these genetic drivers of disease, and they can act through multiple mechanisms that can influence pathology. So we'll walk around what these are, but effectively they can sort of change how genes are expressed, and they're influenced by the landscape of variants around them. What makes them I think sort of this versatile lever, if you think in terms of controlling pathology of disease, is that they can act at the information level pretty much across the entire central dogma of molecular biology. So if you think about what we heard about, is we have the genetic information in the cell, it gets transcribed into RNA molecules. It's important to recognize that happens through this step where there's a pre-mRNA, because it gets spliced, so we can interfere with how this assemblage of information gets put together before you get a messenger RNA, which then goes off and ultimately gets turned into protein. And ASOs or siRNAs or oligos generally can sort of interact at each one of these stages in different ways to have potential therapeutic benefit. When we think about what they are at a basis, they're short by a class. We usually think about them as less than 100 nucleotides long, which is less than 100 of these little monomers that we put together. They're polymeric in nature, and what we need to do is we need to take what is typically considered the endogenous or naturally occurring bases and nucleic acids, and we need to chemically modify them, because the cell, as a general rule of thumb, has evolved to not enjoy having foreign DNA or RNA into its cytoplasm, into its work. So we have a bunch of mechanisms to protect us from that. So we have to think about how do we evade that mechanism so that we don't cause additional pathologies as we treat with these sorts of medicines. So when we think about what that are, I'll just show a couple pictures. If you dig in one step deeper, and this is really part of demystifying it, each one of these polymeric drugs is composed of these units. These units are effectively these nucleotides. Nucleotides have pieces to them. They have these backbones, phosphodiester bonds are what we call them in terms of normal DNA. We modify these sometimes, but you have linkages, you have sugars and nucleobases. And what's important to recognize is that for each one of these modifications that we put into this polymer, you can change any number one of these. So this adds up to being a pretty daunting challenge in terms of how do you optimize these sorts of medicines. And that's the key takeaway message. But when we think about how they can actually impart change or impart influence on the sort of molecular biology of the cell, I'll walk through a couple of the mechanisms in which they gain sort of a molecularly work, and then we'll walk through how those molecular interactions can impart, hopefully, corrections to pathology. So I'll start with one which is a splicing only, and What happens here is you actually bind an oligo, and it binds with sort of A to T, G to C, Watson-Crick-Franklin hybridization for those who think about it. And once you bind to it, you effectively block one of the two isoforms. And I show here, this is the splicing I was talking about, where you have a gene modeled out with exons and introns. Exons are expressed. The E is the mnemonic that you can use to remember that. And effectively, by blending an oligo here, you can block one of the isoforms. So if you have an isoform you want to get rid of for whatever reason, we can get rid of it. That's really handy, so you can correct diseases that are spliceopathies. You can use that to upregulate gene expression sometimes, if sometimes the splicing is effectively driving some of the isoforms to be not expressed or turned into protein. So by blocking that, you create more protein. And there's other things you can do by effectively just binding oligos on a transcript. The other thing that is frequently done is you might bind an oligo directly to an mRNA, and then by doing that, you form what would be thought of as a DNA-RNA duplex. Turns out the cell hates these, so there's an enzyme that sits around. It's in every one of your cells, and pretty much every cell in the body, and every cell in most living things, called RNase H1, and it binds to these RNA-DNA duplexes, and it gets rid of the RNA that's bound to DNA. So you can do that, and that can also lead to destruction of an RNA molecule. That can also be helpful if you have a toxic gain of function or an aggregating protein. What's interesting with single-stranded ASOs is they can work both in terms of the mRNAs, but they can also work on the pre-mRNA. Thinking about the disease biology that you might need to impart on, this means that you can impact things that are often thought about as undruggable, like long non-coding RNAs or other types of regulatory variants that you need to control. So again, the RNase H comes in and blocks. And then the last piece is you have siRNAs as well. They work in a very similar mechanism, but just to be clear, they work by coming in as a duplex typically, and then they eventually recruit a RISC complex, which then kicks out one of the strands, and then that together goes and finds the RNA molecules. Similar story, though. At the end of the day, what they end up doing is blocking the eventual creation of protein by getting rid of that transcript intermediary. The thing to keep in mind with siRNAs versus ASOs is that they only work on mRNAs, and clinical results in tissues outside of the liver have been not yet validated yet. So I'll just highlight, and what I wanted to do here, I'll go through this, and this is obviously something we can talk about afterwards or you can dig into on your own. But each one of these, what's interesting, I have a couple of examples, is that we use these technologies where we can interact at the foundational levels with these levers, but each one sort of has a unique story, and I think that's something that I think everyone should recognize, is that OBMs generally provide this lever to control the molecular biology of the cell, and you can use that to have different in parts on correcting pathology. So for instance, this Vertex program with using a guide, if you will, for, I can't go back any part, a guide to sort of edit BCL11A is really interesting. It actually turns out they're changing the promoter of this, which is a repressor that turns on a fetal hemoglobin gene. So pretty interesting, but they're not editing directly the pathology. They're editing something that's knocking out something that would normally be expressed only during fetal development. And then this one's an ADAR program. This was pretty straightforward. They're trying to edit a variant that exists in the serpin A1 gene. And they can do that because there's an A to I variant that they can correct. But with ADAR, you can only really turn these A's to I's, so there's only a subset of variants you can target. With splice modulating oligos, we have Spinraza that targets SMN2. What's cool about this story, of course, is that SMN2 actually creates SMA protein, but normally we all have this extra copy of SMN2. Most primates do, but it's actually SMN1 that has the pathological variant that causes SMA. And what was discovered was that you could reactivate effectively this gene, this extra copy, with an oligo. Again, it's useful to hear these stories because they tell you that it's not always directly easy to think, I'm gonna change this one gene, that's the gene where my variant is. We have levers, we have tools. How can we use those to impart the favorable effects we want? Clin7, this is Melicin. Temu was able to correct splicing on a variant. This is a very direct one. And then Stoke has this program with SNCA. And again, they're changing splicing, but instead of changing splicing because there's a true spliceopathy, it turns out there's a SNC1A has a transcript that's made that's normally just degraded right away. So by blocking that degradation, they're able to upregulate SNC1A. As we move forward with RNA degradation, SOD1 is an aggregating protein that can just get rid of it. Same with TTR. And then UB3A is another really interesting story where it was actually they needed to upregulate this gene, and it turns out there's a non-coding RNA that's just proximal to this protein that they could then knock down that has an impact of actually upregulating UB3A. So although it's a knockdown mechanism, it has the opposite effect of what you might think. In terms of translational modulation, I'll just say that these are in early stages of work, so there's not much to talk about. And then aptamers, they sort of work as binders, sort of almost like antibodies, but they bind to proteins. And there's a VEGF interactin and C5 that are in development as well. And then the last thing I'll just highlight is as you think about optimizing these compounds, the real challenge is how do we make them so that they bind and they retain safety as well. So, and I'll just highlight real quick that the challenge is that sequence, if you think about 16-mer, we were talking about the number of small molecules that we can screen. If you just think about a 16-mer oligo of ACTs and Gs, if you do the math, it turns out there's 4.2 billion some odd sequences you can write down with just 16 ACTs and Gs. But it turns out it's even worse than that because we have to chemically modify them. So you have to change the backbones, the sugars, and the bases. So as you add those complexities, you actually get to the 10 to 25, 10 to the 30 possible permutations. So my point here is that it's not as simple as just finding something that binds and moves forward. You have to expect there's going to be a translational jump that has to be made to ensure that even though you have activity in, let's say, a cell model, to make sure it's going to be safe and active in an animal and a person ultimately, there's work to be done. So hopefully that brings us back around to our key takeaways, and everyone can walk away with the substrate of information to sort of move forward with, which is, oligos are a little bit demystified in terms of they're just chemically modified RNA, DNA-like molecules. They're simple polymers made out of nucleic acids or amidites, and they work through multiple mechanisms. And by understanding the foundational biology around the gene of interest or whatever is driving the pathology that needs to be corrected, we can think creatively on how to use these levers to impart, hopefully benefit in terms of patient course. So I think I almost made it in 10 minutes. Awesome.
Rachel, you want to talk about mRNA?
Sure. So I can give you maybe a little bit of an intro into mRNA, which is a little bit different than the ASOs and siRNAs. So as you mentioned, Chris, generally with an ASO or an siRNA, you're trying to knock down the expression of something, though if you're knocking down a repressor, that may ultimately result in higher expression of something else. So, understanding the biology there gives you those levers that you can kind of push and pull. But with mRNA technologies or messenger RNA technologies, really what we're doing is enabling the cell to produce a protein of interest. And so, Chris, you had on your slides, cells naturally have mRNAs. They're what serve as the instructions to make proteins in cells. What we're doing with mRNA therapies is really making the mRNA in an in vitro transcription reaction, so in sort of a test tube, if you will, and producing that, chemically synthesizing it, and then taking that molecule, combining it with a delivery technology, and having that mRNA enter into cells to make a protein of interest. And probably people here have had some experience with an mRNA therapy or a vaccine, if you will, in the form of the COVID vaccine. So in that case, companies took the COVID spike protein, encoded that in an mRNA, synthesized that in vitro transcription reactions, so in a test tube, purified it, combined that with a delivery technology. In that case, they were using lipid nanoparticles or LNPs. This helps to shield and protect the RNA as it's delivered in the body, and also enables delivery of that mRNA to cells in the body. So you received an intramuscular injection of the mRNA encased within the lipid nanoparticle. It entered into your cells and it expressed the spike protein of the coronavirus. And this instructed your body to basically produce an immune response, sort of prepared it so that when later you were exposed to the coronavirus, COVID-19, you could more quickly mount a meaningful, useful immune response to protect you from severe infection. And so mRNA therapies basically allow you to produce a protein of interest. In the case of coronavirus, the spike protein. In the case of other diseases, understanding the biology to be able to supply and make a protein that is either folded incorrectly, isn't produced to sufficient levels, these types of things. That's what mRNA technology can really help to provide as a therapy. But just to leave you with the idea that there's sort of two key parts there, the mRNA and the design and making of that mRNA itself, and then combining that with a really useful and effective delivery technology. mRNA is unstable. We're making really long RNAs as well. So to give the cells instructions to make a protein, we're talking about several hundreds, if not thousands, of nucleotide bases that we have to make into this mRNA molecule. So a little bit longer than the 16-mer or 20-mer or so of an ASO or siRNA. And so we don't have as fine control over the chemical modifications that we're making there as well. So some of those can help with stability profiles. Maybe someday we'll get there. Right now, we sort of can change or modify some of the bases that we're incorporating into the mRNA, but just in sort of an ineloquent way, it's sort of incorporated as we're making the RNA as we go along. We can't control specifically where we'd like those changes to occur. And then the other thing about mRNA is it's sort of inherently unstable. It's not meant to stick around forever. In your cells, your DNA serves as the sort of repository of your genes, and mRNA is meant to sort of carry that information into the cytoplasm to produce proteins, and then it's degraded. And if you need more, you make more mRNA to carry those instructions, and those instructions change over time, whether you need more or less of a given protein, and that's how we have sort of gene regulatory networks. When you produce an mRNA therapeutic, it kind of has a very similar profile. So that mRNA can be expected to produce a protein for hours or days, but we're not talking about months and months of expression from this one RNA molecule that we're providing as the therapy. So sometimes what you'll see is that people need to be dosed over and over again with these different therapies to be able to supply that protein over a long period of time. And so, in some ways, that can be a drawback. You have to be redosed. That can lead to high cost of goods. But it's also sort of early days for the technology. And I think one really interesting application of using mRNA therapeutics and sort of this idea of delivering a protein to produce an effect is maybe to use some of these gene editing technologies where you're relying on the gene editor to create a more long-lasting change in the genes, so then that redosing issue is sort of resolved. I think that's where much of the field is going. We're not quite fully there yet, but it's early days for the field. Maybe just to speak briefly to companies that are working on some applications for rare disease. Moderna just last week had a really nice R&D day where they went through several of their programs. Obviously, they're very active in the vaccine space, but they do have several programs in rare disease. It's something they have said they've made a commitment to, and so I would encourage you to check out their website. They have a really nice presentation from that day, and I think the webcast is probably also available for replay from that event last week. They have several programs really focused on being able to supply a protein that's missing that could be secreted and expressed to supply that needed protein. So those programs are given intravenously, and then they have a lot of expression from liver cells. That's sort of one of the maybe drawbacks of our current understanding of mRNA therapeutics is that when dosing using these lipid nanoparticles, they tend to accumulate in your liver. If you think about dosing with these particles intravenously, your liver is there to really filter your blood, and that's just sort of where they get caught up, and then they enter into cells and you have expression in those areas. But using this approach for a secreted protein, may make sense. It's available in your bloodstream, and that can supply the protein that's missing. They also have a program that's partnered with Vertex focused on cystic fibrosis, and they're using an aerosolized approach to deliver the RNA to lung cells. There are several other companies. ReCode has a similar program and others focused on cystic fibrosis, as well as a local company, Arcturus, who's using kind of a little bit different form of RNA for some of their programs. They also have some programs focused on CF and rare disease. So you could check out those websites and learn a little bit more about those programs.
So if you were with us for the previous talks, if you go back to Maya's slide and think about DNA at one end and protein at the other, these therapies are all targeting that midground that's translating the DNA instruction into the protein. There's a very seductive elegance to that. But if you think back, I mean, anyone who's familiar with Gilead, which has become a huge company doing antiretrovirals, they started life as a siRNA company and failed miserably. And it was only that they found these drugs in Czechoslovakia that they thought that they were able to save themselves. But what makes this such a challenging area to produce a therapeutic?
I think in my mind, there's two aspects of it. On the smaller RNA therapies, ASOs, siRNAs, there's been effectively, I think, two large challenges that the field had to address. One is general safety of these compounds, and the second is delivery of these compounds into the right cells and the right tissues. On the safety side, what we've learned over the last, they've been in development for probably since the late 70s, so over the last 40 years or so, is that it's true with any drug, but it's certainly also true for oligomedicines, is that the things that can cause safety issues are pleiotropic. You can cause cells to be unhappy because you put something in that kind of looked like a viral RNA. You can cause cells to be unhappy because you cause two proteins to aggregate that weren't supposed to aggregate. And then when you get into the clinic, you can be surprised because it turns out immune systems of all the non-clinical species you work with, whether it's a mouse or rat, unfortunately dogs and monkeys, those immune systems are also not like human immune systems. So by the time you go into a human, you can be surprised by immune responses. The upside is that over the last 4 years, we figured out how to model many of those things in preclinical ways and in ways in which we can address them early on. So now I think we're in a place where we have a better handle on how to weed these things out. It's still a challenge to understand what is the optimal sequence in chemistry that's going to be the right drug to move forward, but I think we can solve those. And on the delivery side, I think there's been, for single-stranded ASOs, we're fortunate that they can just be absorbed into cells pretty well because they're charged, they bind to proteins, and those proteins get absorbed into tissues, both liver, and then if you inject them into the CNS through intrathecal delivery, they last for a long time, and you can get away with without any sort of additional delivery modalities. But on top of that, we're getting to a place where we have delivery modalities that can target particular cells and tissues, and I think that'll start pushing us over the edge.
Are the challenges the same for mRNA?
I think they're similar. Certainly the delivery issue is also a big one for mRNA. Finding LNPs or different delivery technologies that would allow you to go beyond the liver, particularly for other organs and tissues, is an area of very active investigation by many people. Maybe trying to target LNPs, attaching an antibody or some other targeting moiety to the LNP to have preferential uptake in those cells, so that you can deposit that RNA to have expression specifically in those cells where you do need that protein or you're lacking it, is a big one. And I think for mRNA, just being able to make it was a real challenge at first for people. You can make very small scale things in the lab, but having enough to be able to dose humans in a meaningful way, that was really challenging, and it took some time to develop the technology to be able to synthesize it.
We've now had market experience with these therapies. Where are we in terms of tapping their potential?
For mRNA, I'd say we're very early. The COVID vaccine is, a great example of something that worked, but honestly, when I first saw the efficacy values that they put up, I thought it was a typo. I actually didn't believe it at first. So it's an amazing technology, and I think the pandemic really accelerated the development of the technology. Moderna has said by 10 years or something like that. But really finding the utility of this for applications beyond vaccines is where people are trying to go, but it's still very early days yet. Yeah, we don't totally understand the biology behind every single aspect that we're doing.
Chris?
And I think for ASOs and siRNAs and other sort of oligomedicines, I think we're still at the early days of where they can go. And if you think about one of the exciting things right now is in the post-genomic world we live in, if you think about, even 20 years ago, it was hard to get your genome sequenced. Now, you get genome sequenced, you get it back in a day if you end up in the NICU. That puts a lot of pressure now on sort of how do we actually accelerate translating those genomic discoveries into meaningful therapies. And I think one of the things that excites certainly me and I think a lot of the field is that these RNA therapies actually do provide that capacity to translate very directly the information that you learn from these diagnostic events into therapies that can be meaningfully impactful in correcting disease pathology. But we need to get better at going fast because right now it costs too much and takes too long to make these medicines.
So in that chain from gene to protein, we're moving upstream, upstream. Is your sense that something like gene editing will obsolete RNA therapies, or are they going to be preferable for certain applications?
I think there's a place where gene editing will eventually be very useful. I think there's a long tail of indications where actually having a dosable therapy that you can control and you can have both you can stop dosing or you can have control over how much you dose is going to be very valuable. Even if we had completely precise and perfect gene editing, I think having dosable drugs that you can get into different cells in different ways will have real value for a lot of therapies.
Yeah, I would agree that, you want that modularity to fit the profile of, duration of expression that you may want, dosability, the ability to stop treatment. And then I think mRNA therapies can be a modality that allows you to deliver the gene editing enzyme, for example, that allows that to occur.
So as you look at the world of diseases, what makes one particularly well suited for this type of approach?
Yeah, I guess I'll start, and it's sort of, it really comes down to whether or not there's a line of sight that can be developed between the levers that we have good understanding of how to use, whether or not you're using an ASO to knock down a gene, or an ASO to change splicing, or whether it's an siRNA or an editor, like an ADAR or even a DNA edit. If you can make a connection between that molecular lever you can pull and how it's going to have some sort of impact on driving a corrective event in the cell, then that's very impactful. So in my mind, it's really about making sure you can draw these lines of, what are the things that we have control over, and how will that impart a pharmacological effect that has a reasonable aspect of driving benefit, and can we do that in the cells that matter? Can we get it into the right tissues, into the right cells, in a way where we expect to see a real improvement in patients?
Yeah, I think that delivery piece is really key and is lacking currently for mRNA therapies, but lots of work being done in that area.
If I was a drug developer and I had access to a broad toolkit of therapeutic approaches, when would I use mRNA versus an ASO?
Yeah, so they kind of have a little bit different effects,
Mm-hmm.
but if you understand the biology and you have specific sequence, you may be able to target something specifically with an ASO, and maybe there's a better delivery profile there because just in terms of mechanism of delivery for ASOs, it might be more advanced. I think it really comes down to the biology, understanding. Right now for mRNAs, the duration of expression is short, and so unless you're willing to redose, and then probably in the future something will be developed that allows you to have a better profile. So maybe it will be supplanted by something that's a little bit more efficient. But I think it's just all about the biology and being able to affect it.
So, Chris, you kind of did it fast, but in describing the chemical modifications that are made to turn an oligo into a drug, you talked about something like 10 to the 30 variables.
Yeah.
It's a lot of variables. But talk a little about the complexity of turning an oligo piece of code into a drug.
Yeah, so the traditional way of doing it, and full disclosure, I'm co-founder and CEO of Creyon Bio, and we're trying to facilitate making this process faster and more efficient. But the traditional way of doing it is you have a gene of interest where and we live in a world where if you know a gene that you want to target, you know the sequence of that gene, you also likely know the variants that you're trying to correct. So we can just lay oligos across it using, reverse complements, A to T's, G's to C's. That gives you the sort of base sequence, and then depending on what you want to do with that oligo, so if you want to recruit, let's say, RNase H to knock it down, RNase H, we all know what it loves. It loves RNA DNA duplexes. It's like a little Pac-Man in your cell looking to sort of gobble that up. So if you make those oligos, when they bind, look like an RNA DNA duplex, you can recruit RNase H there. But if you want to change splicing, you're going to have to not make it look like an RNA DNA duplex, because that's not what you want to do. You want to actually just block a splicing enzymes from binding to that RNA molecule. And if that's what you want to do, that's great. But now you got to know exactly where the splicing machine is going to bind and what the impact of having your oligo there. So right now it's very exploratory in terms of doing there. And then once you get to the right sequence, a lot of people then end up going through these, what a lot of people refer to as structure, activity, relationship screens. These come out of small molecule world, and they'll start randomly playing around with the sort of chemistries. What sugars we might put on there, what base modifications, what backbone modifications. But even if you had a single, like if you just think about it for a moment, if your oligo is shorter, I'll do the math, but it's easier, but if your oligo is short, let's say 10 base pairs long, and you only had one modification that you wanted to put, and you didn't know how many you could do, but you could put it anywhere or everywhere in 10 bases, you're at 1,024 possible chemical modifications with 10. For every new base you add in there, and for every combination, it just explodes on you. So even when you're fixed with the sequence, you're still at this daunting task. So right now, it's still a case where there's still a lot of trial and error discovery work that's done. I'm optimistic that we'll get beyond that, and we'll be a place where we can truly engineer these, but that's active work.
So you talked about taking an mRNA strand and putting it in a liquid nanoparticle, which is a vector. Is it more akin to gene therapy, or are you doing chemical modifications to the mRNA so it acts drug-like?
So you can do chemical modifications to the mRNA largely through just incorporating modified bases when you're synthesizing the RNA molecule itself. So maybe a little bit less precise than what you can achieve with an ASO or an siRNA, where you can probably specifically choose which one of the 16 nucleotides you want to change and in what way. So if we're just using a modified base when we're synthesizing the RNA, and one might do that because it reduces your cell's ability to recognize that as foreign and sort of mount these responses in your cells that can dampen translation from the RNA. It can just be incorporated throughout, and so there's a little bit sort of less elegance in that, I suppose, but they are longer RNAs, and so that's how it's sort of, we view that currently. In the future, that could change possibly.
So one of the reasons these therapies are so attractive is not only are they precise, but you can quickly synthesize them. If you see the work on what was done with the vaccines, how quickly companies were able to move. One of the things that a lot of people in this field talk about, and I know you feel very strongly about this, Chris, is the lack of data and the need for data. What kind of data do you need? What can be done to generate it, and how can that accelerate the development of these therapies?
Yeah, so I guess I can speak on two sides. One is, what sort of data do we need for, like, what sort of data do patient advocacy groups and what disease groups need to sort of push things forward to sort of motivate development? And I'll push that and say, anything we can learn to sort of put us in a place where we understand the molecular biology, and sort of, even if you have that and you have line of sight of how you might be able to have a corrective impact on the cell, how do we then connect that to corrective improvements in the clinic, natural history studies? So that data, I think, is really important in terms of pushing forward on making progress in the field. On the drug development side, how do we get to a place where we can make these drugs faster and more efficiently? I think we need to look very carefully about where these drugs typically fail in the clinic or in the preclinical space. And right now, at least for ASOs and siRNAs and other short synthetically created oligos, they fail from safety reasons. Everybody in the room, I could teach you in a moment to write a reverse complement, and you'd be able to sort of start writing these drugs down. The reality, though, is most of those drugs would end up not making their way into the clinic because they wouldn't be safe, or they would be safe but lose activity along the way. So the real trouble is, how do you get that data? And that's hard-earned data that you need to do, and you need to understand how to collect that data. So I think it's really important that you start creating these safety, effectively pharmacology safety databases that sort of lead to having the right span. But because the space is so gigantic, the design space at 10 to the 25, 10 to the 30, if you randomly walk around that space, you're still not going to get anywhere. It has to be very systematic in terms of how you think about creating those data sets. So I think there's a hard path to create, but that's where I think there's ample room for industry to come in and start thinking about how do you create these data in an efficient way so that you can push forward and start lowering the cost and time it takes to create new medicines.
How about within the world of mRNA? Is there a similar need for data?
For sure. I mean, I think it's maybe even a little bit earlier. There's sort of fewer clinical examples of mRNA therapeutics. So, sharing that data as we go along or understanding why things are successful or not will be helpful. I think a lot of the data is being generated in the preclinical space. It's really hard to get access to some of that data. And in particular around the delivery technologies, there's a lot of work being done there because that will really open up the space to be able to have these mRNAs delivered to different organs and tissues. And so there's lots and lots of testing happening there. And again, similarly, it's a huge space. You can wander around in it and probably get nowhere. It has to be a little bit more directed. And so using AI or sharing understandings about what has worked, what hasn't, will help us all to advance faster.
So we touched a little on the way AI is being integrated into the drug discovery and drug development process. I'm wondering if each of you could talk about how you view how that's going to change the work you're doing and how that might accelerate the development of therapies, and particularly to what extent you're using AI today.
Yeah, so maybe I can start since I just mentioned it in the context of screening different delivery vehicles. I mean, if you think about how many permutations you could have and trying to look through, if you just think of all that data in like an Excel sheet, for example, there's really no way you're going to look at that and be like, oh, I see a pattern amongst these different things. I think that's where AI can be really helpful. These algorithms can help you to pull out things that would be useful to look at more directly, and then you can go into the lab and experimentally ask the question, is this impactful in improving some aspect of what we're doing? And then I know the previous speaker had put up an image, and I think that's another area where doing some of these screens where you're just generating a ton of data and looking visually at something, you can see that there's a difference, but training a program that can do that for you and can screen through, it never needs a break, it never, sleeps, it can be running and looking at all these different images or whatever your output is and try to give you an idea of where you want to focus your attention.
Chris, it's a little different for you, I guess.
Yeah, so for the way, I sort of alluded to what we often think about, which is the data space is giant and the design space is huge. This mounts a significant problem because, as everybody probably recognizes, the AI is good when you have a lot of data that's relevant. So if you want to pick out pictures of cats and dogs, you can send it all the images in the internet about cats and dogs are categorized, and you can be pretty confident you can build a machine that'll predict which one's a cat, which one's a dog. When it comes down to making an oligo that's safe or not safe, it's really hard to get that many images or understand compounds that actually have safety. And two, the lines are a lot blurrier and the number of permutations are much harder. So what we've focused on and sort of getting to a place where we can use machine learning AI is how to create the right data set in an efficient manner such that we can feed it into tools. And I think the way that we think about it is that if we focus the AI on a true engineering problem, which is what is the right way of thinking about extracting out these design rules, once you have the right data collected, now you're focusing AI in a place where it really can be leveraged really well, really in a way that it's been meaningfully deployed elsewhere. On the flip side, I'll just say that on the other side of sort of biomarker discovery and then thinking about going into the clinic such that you can sort of mitigate the need for large placebo controls or maybe get rid of them. I think using AI to do virtual twins and other things to have more exploratory biomarkers going forward so that you can understand when you're having benefit without necessarily having to have some large number of patients, I think will be really critical, especially in the long tail of rare diseases, to make sure we can have meaningful trials that can demonstrate efficacy and get to a place where we have approved drugs moving forward.
There's been a lot of interest in the potential to do N-of-1 studies around ASOs, hyper-rare conditions. What's the potential for that, and what do you think we need to do to compress the time to getting there?
Yeah, I think that there's the challenges are the same for an N-of-1 drug, for an 5 drug for an N of 10 drug, which is how do you get to a compound that you're assuredly safe and it has the best chance of having benefit for the patient. I think the challenge is really making sure that, and I'll say also, I just, when you find an N of 1, there's almost always another patient around the corner that you just haven't found because we haven't looked for them. Then you'll eventually get to an N of 10. So I think there's a little bit of a misnomer of an N of 1. It's an N of 1 as of now, but it's likely going to be an N of 10. So I think from a regulatory perspective, the challenge is how do we build out regulatory frameworks that allow us to demonstrate benefit in vanishingly small patient populations, but don't put in some sort of weird step function that disadvantages groups based on the sort of observed penetrance or in number of people that have a particular variant. So the hope I would have is that we get to a place where the regulatory guidance is clear, which is what do you need to actually demonstrate a drug is safe and ready to go into testing in a patient, so you're not going to pass the Hippocratic Oath. You know it's going to be safe and has a prospect of benefit, and then how do we get to a place where we can have a fluid way of demonstrating benefit in the clinic, whether you're dealing with a single patient or you're dealing with tens of patients or thousands of patients. If we get to that place, then it opens up an opportunity for payers to come in and pay for these medicines. And at the end of the day, that's what we all need. We need payers to build the economic pull to allow these drugs to come in, because the last thing I think we want is that, we want, when you have a patient that could be treated by a drug that you could engineer and make that would target a particular variant, you want the prescription to be written, you want the prescription to be filled, and you'd like to go to the doctor and get it made. We don't want to have to go, raise money for it, right? We want to just make the drug, and the way you get that is you got to build infrastructure to make that viable.
We're running out of time here, but before we end, Rachel, is there a takeaway you would want people to leave this session with, and what they should think about or know about mRNA therapies?
Maybe just that it's just very early days yet. We have so few examples of this, but I think there's lots of potential there. So just stay tuned. I think there's going to be a lot of interesting developments in this space.
Chris?
From a future look, I'm excited about the field. I think there's, the world we live in right now as biologists, as people that can leverage the knowledge that we've come out is just so huge. We understand these things. We can dissect the sort of complexity of pathology very rapidly, and we have tools now that allow us to sort of hopefully have levers that'll help with that and provide therapeutic benefit. And I think these are part of the toolbox, and it's just a matter of making sure we connect them.
So we have about 2 minutes for a question. If anyone from the audience has a question they'd like to ask. Well, I'll ask one. We've seen the mRNA vaccines come out at $35 a dose. Is that a function of just making millions of doses? And what's the opportunity to really take some of the cost out of it from the rare disease side?
Yeah, so I'm certainly not a financial or business person. I really come from the research background, but I mean, with any new technology, I think you're going to have fairly high cost of goods, and sort of a lot of the vaccine programs were also very heavily government supported. So I would hope that over time we learn how to make these therapies better, more efficiently. That will drive down some of the cost of goods. There will always be costs that are associated with given research programs, and I guess to your point, if it's spread across a very large population versus a smaller population, there may be some disparities in the price, just because producing a therapy for everyone versus a few people, the quality attributes still have to be there, the understanding of the biology still has to be there, but hopefully the cost can go down over time as we get better at making these things.
Well, please join me in thanking Chris Hart and Rachel Groppo.